All right, so now that we've talked a lot about apoptosis and control of the cell cycle, let's talk a little bit about cancer in general. And we really introduced this in the whole introduction to this unit, so a lot of this has already been done. So I'm not going to get into a lot of the details of cancer. Just a little bit of reminders of tumor suppressor genes, little reminders of proto-oncogenes and so forth, but really just talking about how all of this accumulates. And then the new aspects that I really want you to understand from here are the whole idea of epigenetics also being involved. So it's not just genetic mutations, it can also be epigenetics. And so by the end of this, you should be able to understand what that is and how epigenetics can relate to cancer. And also talking a little bit about cancer stem cells and then the various treatment modalities for cancer. So those will be the new aspects of this asynchronous video. So a few things to think about right off the bat. First of all, cancer is generally considered clonally derived. And so what this means is it starts from a single cell. So remember, these aren't mutations that were inherited. These are accumulated mutations caused by mistakes during the cell DNA replication. And so when the genome is copied inexactly and mistakes are made, that can lead to mutations. And so if one of these cells, all right, so this would be an epithelial cell cancer, but if one of these epithelial cells had mutations that really were in the tumor suppressor gene and the proto-oncogenes or various other ones like we've talked about, that cause it to divide a little bit faster than it's supposed to, that cell, when it divides, the resulting cells are going to have the same mutations, right? And then this is kind of part of that downward spiral, because if that cell is dividing more rapidly, there's a greater chance again for a second mutation. And so then that cell with the second mutation, that's clonal. That's a random mutation that didn't happen in the other ones. But suddenly, if that mutation also causes that cell to divide more rapidly, that cell is going to outcompete even its original cancer cell parents, if you will. And so this is almost like natural selection at a microcosm within your body. And so now this cell with two mutations is dividing faster than the rest, and maybe even starting to be able to divide and stack on top of other cells when it should not, and so forth. And then you can kind of see what's happening here. Then you end up with one of those cells that ends up accumulating a third mutation that allows it to grow even more rapid. And so now this causes some dangerous cell proliferation. It probably would cause some malignancy and possibly even eventually some metastasis. But still, although you do have a few of those original cells with the one mutation, the two mutation, and so forth, the majority of these cells are now clonally derived, meaning from that cell that accumulated that unfortunate set of mutations that really caused those cells, the resulting daughter cells, to be cancerous, right? And so that involves the genetic alterations. So those are the mutations we're talking about. This is mutations in the DNA that causes maybe your tumor suppressor genes to be non-functional. They can't stop the cell cycle, or your proto-oncogenes to become overly functional, and therefore oncogenes always causing the cell cycle. But again, it's these multiple changes, right? Cancer is not a single mutation. It is an accumulation of multiple mutations. So with that in mind, how does that relate to cancer being a disease of aging generally? Well, cancer is considered a disease of aging because it takes time for those multiple unfortunate mutations to accumulate and really cause cancer. So one or two mutations usually doesn't lead to cancer, but one or two mutations can lead to the higher propensity for further mutations, and then you can start to accumulate multiple mutations just like we see here, eventually leading to a cell with the correct or unfortunate combination of mutations that allows them to grow too fast, escape apoptosis, divide on top of each other so they're not contact dependently inhibited, all kinds of things, and even maybe release from the original tumor without losing contact dependent survival signals and go and metastasize elsewhere. All right, so this also is a combination of factors along the way. So you can get cancer because of increased cell division. You can get cancer because of decreased apoptosis, but the reality is it's probably both, a combination of both. So you get increased cell division, along with mutations that prevent them from dying when they should, and now you can potentially have cancer. And so just to kind of bring in a little simplistic analogy for our tumor suppressor genes and so forth, I often relate this to a car. All right, so a car basically has a gas pedal that tells it when to go, but if it's working right, it's only telling the car when to go when you're pushing the gas pedal. So it's only promoting movement, promoting progression when the signal is correct, in this case the signal being your foot. Okay. The tumor suppressor genes then would be like what? Well, the tumor suppressor genes would be like the brakes in the car. All right, and so the brakes are basically stopping the car. And if you think about cancer as being kind of the equivalent of a car crash, anything that's causing the car to go too much and or preventing the car from stopping can lead to a crash. All right, so let's think about that a little bit further. So the tumor suppressor genes, when they're not working, that's like your broken brakes. Right? Now the car cannot stop, just like the cell cannot be stopped as well from dividing. Also, most cars have a set of front and rear brakes, and both must be broken to have a problem. So if just your front brakes are broken, well, your rear brakes can still stop the car, and vice versa. This is equivalent of having two alleles. So since you have two alleles of a tumor suppressor gene, even if one isn't working, the other one can still stop the cell from dividing due to whatever that restriction or that contingency might be. However, if both of your alleles are mutated, then that tumor suppressor gene can't stop the cell from dividing anymore. Just like if both sets of brakes are broken, you can't stop your car anymore. The oncogenes, like I said, are like a stuck gas pedal. This is always telling the car to go, always causing cell division. And even though we have two alleles of proto-oncogenes, if one of them is mutated such that it's causing the cell to, telling the cell to divide all the time, it doesn't matter that the other one's not telling the cell to divide. As long as one is telling the cell to divide, the cell is going to divide. Right? So this is like a stuck gas pedal. Even if you had a weird car where you had two gas pedals, as long as one is stuck, that car is being told to go forward. So tumor suppressor genes need how many alleles mutated to stop functioning? Well, tumor suppressor genes need both alleles to stop functioning because one can still stop, right? How many alleles need to be mutated for the proto-oncogene to become an oncogene and cause cancer? Well, that's only one allele. The difference, and you have to remember this, that mutation has to be such that it's always on. A non-functional proto-oncogene just doesn't tell the cell to divide at all. And then we can now even add in faulty mechanics. All right, so you're much more likely to have a car crash if your brakes are broken and your accelerator is stuck. Then you're really in trouble, right? Well, if you have tumor suppressor genes mutated and you have oncogenes in the same cell, that's that accumulation of mutations that can really lead to cancer as well. And what's going to make this more likely? Well, faulty mechanics are going to make it more likely that your car's not working. And the cellular equivalent of mechanics are p53 apoptosis genes or DNA repair mechanisms. So DNA repair mechanisms, hopefully that's pretty obvious, they are meant to fix DNA if the DNA is broken, or if there's mutations, they can often find these mutations and fix them prior to cell division. So if those enzymes aren't working, well, you have a much higher propensity for mistakes, and a much higher propensity for mutations gives you a much faster chance of accumulating those unfortunate mutations that lead to cancer. All right? Then you add to it that the cell cannot die when it has some of those mutations. For example, p53 is really that sensor that signals that there is abnormal damaged DNA, stops the cell from dividing, gives it time to fix those mutations, and then ultimately helps signal apoptosis if the cell can't be repaired. If that's not working, then suddenly that cell is continuing to divide when it should have been stopped. Just like a car being sent back out in the street when it really has all kinds of problems and should not be. So if you have a faulty mechanic, you're much more likely to have a bad car that can get in an accident. If you have these types of enzymes here, p53 apoptosis genes or DNA repair mechanisms that are broken and not functioning in the cell, then you have a much higher propensity to get those mutations that can lead to cancer. And that, in turn, is the downward spiral. Again, I know I keep talking about this, but I really want to hammer this home. So you can have one mutation that leads to a cell dividing a little bit faster, right? And that is going to continue on clonally, but that might lead to a second mutation that causes even further cell division, and but still not cancerous. And then that might lead to a third one, and then on to a fourth one. And it's that accumulation of mutations. That accumulation of mutations that can give you abnormal malignant tumor cells, ultimately metastasis, and so forth. All right, so next I want to talk to you a little bit about epigenetics. So what is epigenetics? Briefly look it up and see what you find. Well, epigenetics is basically a change in phenotype without a change in genotype. All right, and so this is something that can contribute to cancer even though you don't have an actual genetic mutation, meaning a nucleotide or a gene sequence where a G was changed to a T or an A was changed to a C or something like that that we consider normal mutations. This is something that can cause a phenotypic change, including promoting cancer, even though there's not a mutation in the gene. And it relates to how the genes are being used, how active they are, and so forth. All right? And so this is kind of the normal aspect, the mutational aspect we talked about. So if you have a mutation, an accidental change in the sequence in the DNA, that will get passed on to the other cells. That is a mutation that might stop a tumor suppressor gene from working, for example. However, epigenetics more relates to how tightly wound the DNA is. All right, so let's come back to this in a second. So you've probably heard about this before. You have euchromatin and heterochromatin. Which one of those are genes that are actively being transcribed and therefore actively being used, are on? Well, euchromatin are the genes that are on. Heterochromatin are the genes that are tightly packed away. All right? And so you can think of heterochromatin as that ugly sweater you got from Grandma one Christmas, and so it's not in your normal shirt rotation. It's in a box tightly packed away in the back of the closet, but you can't quite get rid of it because every once in a while you have to pull it out and wear it on Christmas so Grandma doesn't get offended or something like that. But anyway, heterochromatin are genes. They're still there. They're just tightly packed away and not being used. Euchromatin are wide open genes that are available for transcription and are being used quite a bit. All right, so with DNA, so we have nucleosomes. All right, nucleosomes are these proteins that wrap the DNA. So with DNA that is tightly wrapped around nucleosomes, be more or less likely to be turned on, more or less likely to be expressed. Well, if they're tightly wrapped, again, like the analogy with the sweater, that's tightly packed away. So the more tightly wrapped they are, the less they are being expressed. And so epigenetics relates to this. One of the reasons that our cells act differently from each other is not because they have different genomes, but because they're using different combinations of genes. Another analogy I often like to use is you and I, we might have the exact same cookbook at home, but that doesn't mean we're going to have the exact same dinner because we might choose different combinations of recipes, and therefore even though we have the same instruction book, we're having completely different, phenotypically different dinners, if you will, so to speak. All right, so our cells, even though they have the same genome, they act very differently because a neuron is turning on different sets of genes than a muscle cell, which is turning on different sets of genes than a red blood cell and so forth. All right, epigenetics is that change in phenotype, pretty dramatic on different types of cells, even though they have the same genome, so not a change in the genotype. Well, we can have that happen with cancer too. So for example, you can have a gene that should be active become tightly wrapped up. Right? So if a gene that should be active is tightly wrapped around nucleosomes, then that means that that gene is not being expressed. And see if you can think of an example about how that might lead to a cancer. What are some examples of genes that should be expressed in a cell that if they were not expressed, that would lead to cancer? Well, think about our tumor suppressor genes. We talked about genetic mutations that render them non-functional. Right? So if you have a mutation that makes the tumor suppressor gene not work, it's not stopping the cell cycle in situations where it should be. Well, if that tumor suppressor gene is not being expressed because epigenetics are incorrectly wrapping that tumor suppressor gene up, then that tumor suppressor protein is not being expressed, and that's no different than if it didn't work. If it's not there, it can't function, it can't work. So even though you haven't changed the sequence of the gene for the tumor suppressor gene, if you've wrapped it up tightly by epigenetics and it's not being expressed in a cell where it should be, then that tumor suppressor gene is effectively off, right? On the other hand, you can do the same thing with proto-oncogenes. So how might this relate to proto-oncogenes? Well, if a proto-oncogene is normally regulated at the transcription level, and therefore it's only expressed to tell a cell to divide it or to promote cell division when it's being made, if that epigenetics, if that tightly packing away that gene so that it's only on at certain times is not working and that gene is open and always on, well, now you're making a proto-oncogene at all times. That's similar to an oncogene. It's therefore always there telling the cell to divide, even if it's not mutated by a sequence. Right? And this epigenetics basically relates to wrapping around histones. The tighter wrapping around histones, the more closed, compact, and therefore inactive that gene is. But this also relates to DNA methylation. Right? And all of these things are things we talk a lot about in developmental biology. So if you're taking developmental biology, we'll talk all about how epigenetics really controls development, differentiation, and even evolution in some ways, right? But we'll talk about epigenetics, but DNA methylation also can cause a gene to be tightly wrapped and hidden away. So we can do this at the protein histone level and at the DNA methylation level. But either way, if the epigenetics is messed up such that a gene, a la tumor suppressor gene, is hidden away and off when it should be on, or a proto-oncogene is not hidden away and therefore is being expressed when it shouldn't be, that it can lead to cancer just as much as if there was a genetic change in the sequence. That's how epigenetics play a role. Okay. Next, we have cancer stem cells. All right. Well, stem cells are able to divide indefinitely because they basically turn on telomerase. And so there's a theory that only stem cells kind of remain in the body long enough to accumulate the wide number of mutations. So oftentimes it's actually the stem cells and not the somatic cell that really causes cancer. And you can think about this with regards to skin cancer, for example. All right. We have epithelial stem cells that are constantly dividing and regenerating the layer of skin cells that we often slough off and lose over time. So that stem cell is constantly dividing. If you mutate the outer layer of your skin, the epithelial cells, well, they're not going to divide there very much at all, and they can't accumulate those mutations. But if you mutate the stem cell that's constantly dividing and making all of your outer cells, well, that stem cell, once it's cancerous, is going to contribute to cancer a lot more, right? Because those stem cells are already self-renewing, they already have telomerase activity. In a way, they're already just naturally and by their function halfway towards a cancer cell if you think about it anyway. And that's not saying they're doing anything wrong, but they are constantly dividing already, and so it doesn't take as many mutations to get them to a point where they might be cancerous. This also can be a good thing. This might be one of the reasons why metastases are rare, because if you have a tumor cell itself that branches off and goes, if it's not the stem cell, it might not actually be able to establish a new tumor elsewhere. Right? So within a population of cells taken from a tumor, there's usually a small population inside it that is actually the cancer stem cell. And this is a very small population. This would be a flow cytometry. This would be the cancer cells, and then there's this small population here of stem cells, but that population can make it the cancer continuously regenerating. So you can basically have a normal stem cell that's self-renewing, and then you have a transient amplifying cell. So this is the cell that's being differentiating towards the mature cell there, and along the way you can get mutations, epigenetic changes, that kind of cause it to become cancerous. But if that cancerous is also able to do self-renewal, that's part of cancer. And so as those cells are growing, they will still promote and make all of these other cells like they normally do. They're just making them at a fast rate, right? These are the cells that are basically differentiating. They're just making them a lot more. But inside that tumor are these stem cells. And these are problematic for a couple of reasons. One, they're very hard to get rid of. And two, they also can continue to accumulate those mutations like we were talking about. Right? Stem cells divide much more slowly. Seems counterintuitive, but their self-renewal is much more slow. They actually are making a lot of the other cells that their job is to kind of make those mature skin cells, etc. So they're making those more often. So their self-renewal, their own division, is a lot more slow. And so why does this make them less susceptible to cancer treatment? What do most cancer treatments actually accomplish or do? Well, most cancer treatments basically kill rapidly dividing cells. So if you have a tumor, most of these cells are going to be rapidly dividing. So even the differentiated ones that shouldn't divide, they're starting to divide. And so that chemotherapy may kill all of your cells here, kind of shown in yellow, the differentiated ones. But they might miss a lot of these slower dividing stem cells, cancer stem cells, and if you leave those, even just one or two of those cancer stem cells that are left, they can basically remake the tumor over time. And that's why if you're treating cancer, you pretty much have to get rid of all the cells. Any cancer stem cell left over really can cause the cancer to come back, whether it's a few weeks later, a few months later, or even a few years later. All right. The other thing I want to talk about is really the cancer microenvironment, the milieu. So the cancer cells are only a small part of the whole tumor. The cancer cells are going to hijack the body's normal systems. And this is full circle to what we talked about in the original introductory aspect of this. So the cancer cells are going to remodel the extracellular matrix, which can alter the way the cells behave. They're going to hijack the immune cells. They're going to get rid of those M1 macrophages that would normally recognize abnormality and kill those cells, and basically hijack them into M2, which are ones that help a cell divide and grow and so forth. There's a lot of growth factors that are in the extracellular matrix that cancer cells can cause to be released, thus stimulating their own division further. And they're also going to hijack the blood vessels. They need to create a blood vessel system to feed the rapidly dividing cells with nutrients, glucose, etc., and also with oxygen and so forth. So this whole kind of milieu of cancer cells and all the cells they've kind of hijacked to do their bidding, this is all part of the tumor. All right, so let's last talk about cancer treatment methods. So what are some cancer treatment methods you can think of off the bat? Well, we've already talked about chemotherapy. This is the most commonly understood one. These are chemicals, drugs, that basically kill rapidly dividing cells. The benefit is tumors are rapidly dividing cells, so they can actually be quite good at killing cancer. The problem is that it's not very specific. Usually chemotherapy is delivered systemically, so intravenously, and so now that has access to your entire body. And unfortunately, tumor cells are not the only rapidly dividing cells in your body. So let's think about what are some of the major side effects that you know to be associated with chemotherapy. Well, most people realize loss of hair. Well, your hair follicles are kind of the stem cells at the bottom there. Those are rapidly dividing cells, and so that's why you lose your hair. Nausea. Well, the epithelial lining of your stomach and digestive system, those are rapidly dividing cells. We talked earlier about how epithelial cells are not only exposed to carcinogens, but also are dividing very frequently and rapidly, and so therefore have a higher chance of accumulating the mutations that lead to cancer. Well, that also means that they're more susceptible to chemotherapy, which is why during chemotherapy people are very nauseous. Immune cells are rapidly dividing, so people undergoing chemotherapy are very immunocompromised. And this is one of the huge issues right now with COVID-19, because people that are immunocompromised that's one of the key secondary problems that can cause COVID-19 to become lethal and so forth. Another one is radiation therapy. All right, so this is use of high energy particles, radiation, to destroy cells by extensively damaging DNA. So this is basically taking your cancer cell and realizing that most of the time those cancer cells are becoming cancerous because of massive changes or problems with the DNA. And so if you cause way more excessive damage to the DNA, even tumor cells can't live and grow, so you're trying to kill them in that regard. The benefit is, again, you can kill tumor cells. It's also a lot more localized, so you can kind of use the radiation gun, as it's often called, to try and target the cancerous cells. However, it's still not completely specific. So anything around that, or anything like the skin on the way into that tumor and so forth, is going to be hit by that same radiation, and it can lead to some pretty massive burn problems, and also mutations in other cells around it and so forth. So it's really only effective near the surface of the body, and depending on how they're doing that radiation, it's even though it's a little more targeted, it's certainly not highly targeted. And think about what they did to Henrietta Lacks, where they put that radium basically inside her cervix, and the radium just secreted radiation all around. Well, that may have done something to the cancer, although hers was so aggressive we'll never really know, but it probably did a lot of damage to Henrietta Lacks and her reproductive system as well. And then last, kind of more new, but this relates it to my own research, or one of the projects I do, is angiogenesis. So this is the growth of new blood vessels from pre-existing capillaries. So the interesting thing here is this process in the normal adult is limited to wound healing, menstruation, and pregnancy. And pregnancy being the big one to think about. So obviously, newborn or a newly developing fetus needs a lot of blood vessel growth as that body grows. So you wouldn't want to block blood vessel growth if someone is pregnant, because that would really hurt the fetus. However, again, in normal, healthy adults that aren't pregnant, this is highly limited. And so it really only happens when this process is hijacked by cancer. So after development, the blood vessels you have are really the ones you keep. But if you think about it, what a tumor is doing, it's rapidly dividing cells. It's going to eventually grow too big for the oxygen and nutrients from the established vascular system to really feed it. And so in order to continue to grow, it basically has to cause new blood vessels to grow inside it. And it does this basically by accumulation of mutations. So these cells normally wouldn't secrete the growth factors and the chemokines that cause blood vessels to divide. So these blood vessels are just doing their job. They're responding to signals that are telling them to grow. And these are signals that are really important during development. As you grow, your cells become hypoxic. They're not getting enough oxygen, so they send out signals to cause new blood vessels to grow in there. Well, the hypoxic tumor not getting enough oxygen, especially if there's certain mutations that cause these genes to be on in these cells, they might send out these signals and cause blood vessel growth in them. And it's actually been shown that if you can prevent this blood vessel growth, you can starve the tumor and limit its growth to about that of a small p, the oxygen diffusion limit. So a tumor that cannot stimulate new blood vessel growth can't grow bigger than a small p. And it doesn't metastasize, and there's a lot of things that are better for it. And the great thing is, if you do block this blood vessel growth, you're starving the tumor, but again, since this angiogenesis process in the normal adult is highly limited, you're not really going to have the same side effects, because this is really only happening in the tumor region. And the other thing that's really beneficial to this in theory is the mutations are in the cancer cells. So that's why cancer cells are so heterogeneous. One person's cancer is going to be different than another person's because of a different accumulation of mutations. However, the blood vessels aren't mutated. They're just responding to chemical signals like they're supposed to. And so if you can figure out a way to block this new blood vessel growth, it really could apply to almost any tumor, because the blood vessels aren't mutated, the tumor is. So if you can block the blood vessel growth, the interaction of these cytokines in here, then you can prevent that. The problem is it really doesn't work, or hasn't worked in the past. So if you find something that causes blood vessel growth and you block that one factor, well, there's 100 other factors that can take over. And this makes sense evolutionarily, because if a single mutation could cause you to lose all of your blood vessel growth, that obviously would be very detrimental. You wouldn't live past the embryo because the embryo couldn't grow new blood vessels. So there's a lot of backup compensatory mechanisms. If one goes wrong, another one will take over. And the tumor can hijack those as well. So what we see here is a rat brain tumor. And this one's just treated with PBS, so this is the control. And you can kind of tell the tumor because blue are nuclei. So the cell nuclei are shown in blue, and the tumor is tightly packed nuclei. So this dense part towards the center here in some of these little extensions, that's the tumor. This would be the normal. And the red is a stain for blood vessels. So this is immunohistochemistry. Well, if you block with an integrin antagonist, right, so integrins are needed to help the blood vessels migrate. If you block with an integrin antagonist, well, this tumor still has a large blood supply. If you block with a couple of other factors, this one's blocking the vascular endothelial growth factor, so this cytokine that stimulates blood vessel growth. Well, none of these single monotherapies are effective. However, if you put them together at the right combination, in this case with all three, here you can see the tumor with our control, right, the blue tightly packed again, and you can see that this tumor is highly vascularized. But this is the one where I hit it with all three, triple combination, and you can see that that tumor has almost no blood vessels growing in it. So even though none of the three were effective individually, when you put the three together, you get synergistic exponential activity. And the cool thing is now you can stain for a cell division marker. And this is one of those cell division markers that we talked about. This one happens to be Ki67. It's a protein that's only on when the DNA is replicating and the cell is dividing. So it's only expressed then. So if we use that as our antigen and have antibodies with green fluorescence on them against Ki67, you can see that when you have a tumor that's vascularized with red, it's highly proliferative, lots of cells dividing. If you have a tumor that doesn't have blood vessels growing in it, there's no cells dividing anymore. And eventually, those tumor cells will actually start to die off, and you can basically protect from death by brain cancer in this case. All right? So in conclusion, what we've shown, and we're just trying to find the right combinations, combination angiostatic therapy can basically prevent that backup compensation mechanism. So even though monotherapies are ineffective, if you have the right combinations, you can get this synergistic, really powerful activity. Right? So this is a potential treat for cancer with reduced side effects. The problem is that a lot of the drugs that we tested are not yet approved for market, and they may never actually be approved for market. So it was great proof of principle, but not yet ready for prime time. So we needed a fast and easy way to screen for drugs that are already approved that might have the same synergistic combination. Because once they're already approved, doctors can actually just put them together almost immediately, and they might be useful for treating cancer. So the assay we used is this chick-cam chorioantylic membrane model. We create these little tumors by growing tumor cells in culture and embedding them in collagen, extracellular matrix, and then we just put these tumors on this chick vasculature system. So normally these cam vessels are the vessels that grow around the outside of the shell, or just underneath the shell, I should say, sorry. And that's for gas exchange. But if you take the chick embryo out of the egg, we can actually grow these chick embryos from the time when they're just this tiny little heartbeat to when they're a full-fledged, almost full-fledged chick. And if you want to do that, take developmental biology, because we will actually do that in developmental biology. But we don't actually give the chick cancer, we just put these tumor cells on top of the cam vessels. And these tumor cells do what tumors do. They send out cytokines that tell the blood vessels to grow and branch and form blood vessels up into the tumor. And then we can add various drugs and see if we can prevent that from happening. And more importantly, add drugs in combination. And this is one of the most recent results that we have. So here we have a cancer on plant, and we can then just inject these vessels with a dye that allows them to fluoresce so we can quantify how many vessels are there. So in a control, no tumor one, you get a few vessels, right? So we can quantify the amount of that is vascularized, that is fluorescent. This is the negative control. The 9L is a glioblastoma tumor. And so when you have those cells, you get a lot of vascularization. We then used certain drugs that are already approved on the market, one Avastin, one Toracel. And when you put those individually, you see no difference. You still have highly vascularized tumors. However, when we put this combination together, the amount of vasculature was right back down to the levels of PBS. And so this combination actually seems to be working. And after a lot of times testing various combinations together, this is the one that seems to be working, and we're working on publishing this at this point. And it's highly significant. The increase in vasculature by the tumor is highly significant, and no reduction by Avastin and Toracel, and really importantly, highly significant to the combination. So this is very significantly inhibiting those blood vessels from growing. All right, so thanks for letting me talk a little bit about my own research in this regard. Hopefully the whole idea of kind of combining and applying what we've learned about the cell and its functions to what happens to cause cancer, hopefully that's been interesting and kind of brought this semester around to a pretty cool full circle. So it has been a true pleasure teaching you in class. I appreciate everyone's adventure spirit and their willingness to work with me in this strange semester where the format's a little bit different than normal, but you guys have been troopers and really done an amazing job, and again, it's been a true privilege and honor to teach you and get to know many of you a lot better. So thank you. Good luck on the final exam, and then have a wonderful Christmas break. You all have earned it. All right, thanks.